Good morning and good afternoon to all of our attendees. Thank you for joining today's Family Office Network's webinar series, Scientific Breakthrough, Predict, Prevent, and Cure Cardiovascular Disease. Presenting for us today is the executive team from Arterez. Dr. Joe Tunac is a medical scientist specializing in the discovery and development of drugs. He studies the biochemistry and physiology of the human body and designs drugs to target dysfunctional mechanisms. He discovered his first drug at the Wakeman Institute. He was then hired as a senior scientist and became instrumental in the discovery and development of multi-billion-dollar drugs. He was also awarded the 2015 Nobel Prize in Medicine. Brett has spent his last several years serving entrepreneurs and business owners with strategy and implementation of business development plans, corporate finance, capital raising activities, and mergers and acquisition advisory services. Brett gained his corporate finance, mergers, and acquisitions experience as VP in the investment banking group for Rodman and Renshaw in New York and Chicago, Senior Associate in the Dean Witter Reynolds Corp Finance Group, and as an audit professional for Touche Roche & Co. in New York. Michael Brennan, Chief Executive Officer, is also joining us today. Mike is a serial entrepreneur and business leader with a 30-year record of leading and advising startup, growth ventures, and turnarounds, including 4 startups he founded and grew nationally. Mike's education and certificates include the University of Detroit, Western Michigan University, University of Detroit Mercy, and the University of Michigan. He also has private 501c3 boards and community work that include Detroit-based startup advisory, public speaking for students, tech entrepreneurs, small businesses, and nonprofits. At the end of today's presentation, we will have time for Q&A, and we encourage your questions. Please use your questions chat option on the right side of your screen to send over all questions so the team can answer accordingly. Gentlemen, I will let you get started.
We're ready, thank you. All right, I'm gonna start the presentation here for us today. Thank you all for joining. We appreciate your interest in Arterez and taking the time today to learn more about us. Lindsay, can everyone see me okay?
Yes, you look great.
Okay, well, great, thank you. So first of all, what I'd like to share with you is a little bit of history and background for your understanding. Cardiovascular disease is the world's leading chronic killer, impacting more than 1/3 of the world's population. U.S. direct and indirect healthcare costs due to CVD are expected to grow to an estimated $1.5 trillion by 2030. Among the top 150 developing nations worldwide, CVD is the largest health risk in cost, while available therapies are palliative at best. Statins remain the most widely prescribed drug, with more than 400 million prescriptions in the U.S. alone in 2019. Yet CVD disease and deaths are on the rise. These are staggering numbers, yet medical innovation has practically been non-existent in CVD for the last 50 years. Real innovation is now a necessity, and patients and doctors are demanding it. Dr. Tunac, president and chairman of Arterez, has discovered the multi-factorial upstream etiology or root cause of chronic vascular disease, and that those can be characterized, cured, and ultimately prevented. The company's first drug, Embotricin, targets healing at the cellular level rather than targeting symptoms as most drugs do. The company has also developed a biomarker panel for treatment monitoring, we call it GlycoCardia, that can also be used for disease identification, staging, and classification. It is the first of its kind. As Lindsay shared with you, Dr. Tunac received his PhD from the Rutgers Wakeman Institute, which is the World Center for Antibiotic Research. He was the first student to develop a drug at Wakeman known as hydraheptin, which was an antifungal antibiotic. He then led development of arvamectin for Merck, which Lindsay shared with you, was a Nobel Prize winner in 2015, and later moved to Park Davis, where he continued successful development across the spectrum of disease. For clarity, Joe directly didn't win the Nobel Prize. The drug won the Nobel Prize, and Merck for that development that Joe led. In time, his novel ideas inspired him to become an entrepreneur drug hunter. Soon after Joe began working as an entrepreneur, he developed mitomycin, an anti-cancer drug licensed and sold to Otsuka, which is a Japanese biopharma, for close to $1 billion, which enabled him in 2012 to focus exclusively on the development of vascular research and the development of anti-thromboembolic drugs or compounds for vascular disease, aided by diagnostic biomarkers that Joe also discovered and put together. To understand how best to approach solving vascular disease, which is the single largest disease in the world, Dr. Tunac built a substantial knowledge base of medical science in great detail. He understood that CVD was the leading cause of death, that cholesterol reduction and palliative symptom treatment have been the prevailing standard of care for more than 50 years. He began with an objective, thorough study of vascular disease and the prevailing war on cholesterol that revealed cholesterol is in fact essential for good health and well-being. His subsequent research and development led to this breakthrough discovery that you're going to hear today, that we believe has profound transformational opportunities that could potentially transform the standard of care. Arterez is a Delaware C corporation that wholly owns 2 separate LLCs for the concurrent development of the diagnostics that I've shared with you and our therapeutic technology program. It's important to mention that GlycoCardia and Embotricin are the first products in these categories, but there are many more to follow. I'd like to first, before I turn it over to Dr. Tunac, quickly provide some context for our science. The endothelial glycocalyx is at the core of our science and Dr. Tunac's discoveries. Number one, the endothelial glycocalyx is the fine, slippery lining of the arterial system that enables good blood flow. It houses hundreds of molecules, each with its own function and purpose. 2, plaques in humans are formed underneath this lining, and that is not reflected in the animal models used in the study of atherosclerosis. 3, the majority of cardiovascular disease occurs in the coronary bends where there is a whirlpool effect. And 4, the result of this is inflammation and oxidation, which is at the core of the disease progression. And so I'd like to, at this point, turn it over to Dr. Tunac, who will share with you much more about the science in this breakthrough.
Good morning, folks. Good afternoon if you are in the East Coast, I guess, right?
West Coast.
And good morning if you are in the West Coast. Okay, that's me. What I'm going to do, folks, is I'm going to give you an overview of who we are as human beings. And then I'm going to give you a hint of why we have diseases. And the next thing we're going to do is I'm going to go and tell you what we have done to cure disease. So let me start with who we are. We are 40. What happened here?
You can't touch the screen.
Oh, I lost you guys. Okay. Who we are? Oh, I thought that's okay.
Yeah.
Who we are is we are built of 40 trillion cells, and these cells have to be nourished every day. And here is the basic composition of the cell. You have the nucleus, which is the blueprint of life. And this is the cytoplasm, which is the liquid part of the cell. And this is the endoplasmic reticulum. Bear with me, folks. The technical people would understand a lot, but just follow what I'm trying to do here. The endoplasmic reticulum is the manufacturing site of proteins, and the mitochondria is the powerhouse of the cell. You have about 30. Think about your car. You got 2 pistons, 4 pistons. You got in the cell. You got up to 30,000 pistons that are generating energy. And here's the membrane. Look at the membrane. Focus on the membrane. The membrane is made of 40 to 80% cholesterol. What is unique about the membrane? The membrane defines us as humans or animals. What differentiates us from animals, from the plants, is the plants have no membrane. They have what they call a cell wall, which is rigid. So the 40 trillion cells individually wrapped in cholesterol membrane. We are 9% solid cells, the bones, and we are 91% liquid cells composed of red blood cells, platelets, lymphocytes. The cholesterol is the cell's building block. It maintains the membrane integrity. The brain is 70% cholesterol. The bile is cholesterol. It's needs for our digestive system. Vitamin D is cholesterol, used for immunity and calcium metabolism. Steroid hormones are cholesterol. It delivers messages and activates other organs. The cortisol is cholesterol. Cholesterol is needed for cell growth and repair. And then what you're seeing in the mainstream media is cholesterol. Well, cholesterol is delivered to cells in LDL package, but what you hear in the mainstream media is LDL is a bad cholesterol. In our book, cholesterol, LDL is a good cholesterol because it's the packaging that delivers the cholesterol for the actual use of the cells. So drug treatment to lower LDL is a misguided hypothesis. Here we are. The cells die and replace every day. The only cells that don't die are the neurons. The neurons are the brain cells and the cells of the eye. You are not the same person today as yesterday. The only thing that gives you connection is the brain. So you can see that our cells changes every day, and the movement of electrons maintains life. Electrons provide the electricity. Electrons provide the bonding of the atoms to make the biomolecules. And where does the electron come from? The electron comes from the food that we eat. The food has to be oxidized to release electrons from hydrogen. Hydrogen is the basic fuel of the universe. Even the sun burns hydrogen to provide energy to everybody. So the oxygen moves electrons. So we call the oxygen the breath of life. But the oxygen also, if you have too much of oxygen, it corrodes our life. And we corrode our life starting at 40 years old. We started to go downhill. Here is the mitochondria. I told you before, it's the powerhouse of the cell. Some cells are not very active, but the active cells, like the liver, the kidney, the heart, the brain, the parietal cells, are very active organs, and so they need a lot of power. So sometimes they have 2,000 to 3,000 mitochondria. If you're feeling low and spent, so to speak, your mitochondria, your powerhouse is not working well. So let's go ahead and look at how we produce energy. These are the milk. You got the French fries and the whole stuff there. What your body does, it extracts the hydrogen from those food, and the hydrogen would be stored in a storage tract, which is called the NADH. Sometimes you call this NADH vitamin B3 or niacin. If you feel a little down, take niacin, and believe me, you're snap out of it. You'll have a little burst of energy. So what happens is the NADH trap the hydrogen and deliver it to the mitochondria. And the mitochondria is going to separate the hydrogen, extract the electrons from the hydrogen. So look at the electrons here. The electrons are going to be placed in a tube. Call it the electron transport system. But the electrons are going to stay there. They don't move until you breathe oxygen. The oxygen is the only atom in the world that is always looking for electrons. So if you breathe, it would attract the electrons like a magnet, and it moves the electrons. When the electrons move, you got electricity and you got power. And so the oxygen pulse, and so the byproduct of the electron movement, electricity, is power, called ATP. And 1 ATP is equivalent to about 7 calories. And so the byproduct of all these things is you got water. So you go pee, that's water. And you got to exhale, and you got carbon dioxide, and it goes to the lungs. That's how we live. But for some reason, you have a glitch in the tube, the electron transport system. You get leaking of electrons. When you have leaking of electrons, you create more reactive oxygen. And these are the electric oxygen species here. And collectively, we call them reactive oxygen species, or ROS. And if you eat well, prudent eating, you produce ROS who are beneficial. But when you overeat, you got excess ROS, which is harmful. It comes back to you and oxidates the cells and corrodes life. So how do we age? How do you create disease? Diseases are caused by excess ROS. And how do we get ROS? Well, let's go back to the normal eating. The normal eating, they produce residual ROS, and you could see the list of them. And you have 1.35 to 10 to the 4 ROS per day. This is a normal output of normal eating, because this output here would be taken care of or neutralized by what you call built-in antioxidants in your system. And these antioxidants are you could see it, superoxide dismutase to glutathione reductase. So there is homeostasis there. And you could see the processing speed of this enzyme, very, very fast. But when you are exposed to environment and you have pollutants, you are drugged, wrong drugs that people prescribe in you, you are being stressed out. Your habits, your work activities, your dietary cultures would give you extra ROS. And if you have that, as Mike said, you're going to have inflammation. And this is the beneficial level, but you have more than beneficial level. You're going to have a sequela of diseases. You got cardiovascular disease. You can see that. And the other thing that happens is you are going to age. I told you before, after 40, you start aging. Senescence organ. And one of the things that gimmicks that people are telling you is you have to limit your calorie. Limit your calories, so limit the amount of ROS produced. It's by a starvation diet. The Seventh-day Adventists, I don't know if you're familiar with them, they have a restrictive diet. Sometimes they starve themselves, and this is why they live longer. Vegetarians have the same principle. They try to go ahead, eat vegetables so they don't get a lot of calories. The nuns, they meditate. They have a very serene life. They live long. So the Russians, by the way, they thought about the aging process. And so one Russian guy, this guy's name is Ignatowski, and he thought that feeding milk, meat, eggs would facilitate the aging process. And so he used a rabbit as a model. And then his student named Aneskov repeated the experiment, but he used only egg yolk. And what he did is he observed some fatty streaks in the artery, and he called it atherosclerotic lesions. And this is the birth of the cholesterol hypothesis. But think about the model that they use. They use a rabbit. What is a rabbit? A rabbit is a herbivore. A herbivore cannot process meat, so they accumulate in the artery as fatty streaks. But think about the carnivore. The carnivore could eat all kinds of meat, and they don't develop any fatty streaks. They don't develop atherosclerosis. On the other hand, a carnivore is not used to eating carbohydrates. So if you feed them carbohydrates, they become obese. They become diabetic. Omnivore, like us humans, we can process cholesterol. They don't accumulate in the diet, in the arteries. But in the 1920s, we have this problem. We have the Industrial Revolution, and people become sedentary. We have an increase of CVD. And at that time, the American Heart Association was also born. And they looked at the work of this Russian, and they said, they came up with the cholesterol, they embellished the cholesterol hypothesis, and they used the cholesterol hypothesis as a war to build war on cholesterol. And so in 1961, the AAH kept building up. The organization is getting bigger, so they have more influence. So what they did then is they said, look, we recommend to you not to eat 3 eggs per week because the yolk has a lot of cholesterol. Think about it, 1961, all these years, 40-some years or more, we've been brainwashed that cholesterol is bad for you. But look, think about it, about 3 or 4 years ago, they reversed that. They said you could eat all kinds of eggs at this point. And look at what happened at that time. They almost killed the egg industry. Very terrible. So in 1974, there's a Japanese guy named Akira Endo. He jumped the gun because he eliciting all this info from American Heart Association, and he decided to come up and screen for a drug that lowers cholesterol. He came up with the first drug called compactin. It works in a rabbit, but when they tried it in regular animals like the mice, the dogs, chickens, all the animals you could think of, it didn't work. It didn't lower the cholesterol, so he dropped it. Besides, compactin is a toxic compound, so he dropped that. So what happens? Merck was still in the idea of copying the Japanese, and they came up with a second statin called lovastatin. And at the same time, the government had a hearing. The influence of American Heart Association, they lobbied that cholesterol is bad for you. So basically, the government decided to have a hearing. And they came up with a plan that indeed cholesterol is bad. And so the government decided to have a dietary goal of limiting cholesterol. They came up with a dietary goal without any clinical evidence. So Merck, I told you, Merck discovered the second statin, lovastatin. They tried it in mice and rat. It didn't work, so they dropped it. But look what happened here. There's this guy, Goldstein and Brown, who won a Nobel Prize because of their work on familial hypercholesterolemia, call it for short FH. So they begged Merck to try lovastatin against FH patient. And look what happened. It worked. It was active in lowering cholesterol of patients with genetically disrupted with cholesterol metabolism. And so it lowered the cholesterol. So the FDA approved lovastatin against FH, and voila, the government had a smoking gun, that they could really come up with a drug that lowers cholesterol. And so they, by default, they prescribe lovastatin to everybody, to the public. And so the NIH came aboard, they came up with the adult ATP guideline, and they mandated every cardiologist to prescribe lovastatin, but they didn't know how to measure the effectivity of lovastatin, so they came up with the lipid panel, which everybody knows. And from then on, the fire caught on. And so everybody decided to come up with their own version of statin. You got pravastatin, simvastatin, and the whole works that you see there. But then when they did a clinical trial, in 2004, they have an expanded clinical trial, Mevacor. That's the clinical trial, FAC, pravastatin, atorvastatin. What did they come up with? Statins are not effective against CVD. In fact, the side effect of statin is killing people. 41% between 1990 and 2013 actually had side effects. And so what happens? The FDA issued a black box warning that statins create diabetes 2, liver damage, prostate cancer, memory loss, and dementia. Where does Alzheimer's come from? How come we have a spike of Alzheimer's? We never heard of Alzheimer's before this statin came about. And to me, that's one of the problems of statins. And look, in 2018, the AAH and NIH guideline actually defied the FDA. Instead, they went the other way. They decided to update the guideline, and they said you need to be more aggressive in prescribing statin. And they came up with a new drugs called the PCSK9 inhibitors, Repatha and Prolan. And they said you need to combine statin with this to create lower LDL. And even the president of AAH, he came out and said, no target for ideal LDL cholesterol levels, but guideline recognizes in principle that lower is better. Now, let's get out of the cholesterol problem there. The real problem of diseases is because of the environmental stress. Mike told you that 90% of disease comes from the environment. It's not cholesterol. Environment stress triggers CVD, not cholesterol. So here is on the membrane. The membrane I told you before is made of cholesterol. And here is a seal, the glycocalyx. And if you are exposed to this pollutant, this environmental stress, you get radioactive materials, you got particulate matters, and you got the sun even, and the German pesticide. What happens? These are going to give you additional ROS. You could see that. And this would disrupt the glycocalyx, and then all this reactive oxidase species, they get into the cell, the DNA, the protein, and they disrupt them, they oxidize them, and they become foreign molecules, and they become antigenic. And when they become antigenic, what do? They create inflammation. So let's look at the glycocalyx physically. The glycocalyx is the first layer of protection in the cell. And if you look at the molecular level, there's a lot of ornamentations coming up through the glycocalyx. Here are them. And this is the glycocalyx here. If you look at the glycocalyx is a negatively charged molecule. You have heparan sulfate as the most negative charged molecule in the body. Now imagine this. You have a blood vessel lined with glycocalyx, and the glycocalyx is negatively charged. You have the red blood cells also lined with glycocalyx and negatively charged. Why do you think the blood flows? Because the negative charge of the glycocalyxes of the or red blood cells and the blood vessel repel. That's why you got blood flow. If you disrupt the glycocalyx in either one of them, you're going to have a positively charged, and that would slow down the flow, and you're going to have hypertension and other disease. So the negatively charged glycocalyx is also good because they serve as a nest for all the proteins in your body. They have an example of those beneficial proteins would be a anticoagulant, superoxide dismutase, you got the lipopyrin lysase. So the glycocalyx disruption is the upstream cause of cell dysfunction. Disruptive sequela is manifested downstream and is expressed as diseases. For CVD, thromboembolism is the sequela that produce clot. Clot is the fatal component of CVD. So we need to focus on how to cure CVD, and the focus would be to come up with a drug that prevents clotting. And that is the focus of what we're trying to do. So the quest for anti-embolic drugs is to understand what are the targets we're going to come up with. And I came up with a chart here. So look at the blood vessel. Start with the blood vessel. If you have trauma, if you go to surgery, if you got punctures, laceration, you're going to have clot. That's one place. And if your blood flows into bifurcation, look at the coronary of the heart. You got a lot of bifurcations there. You got a lot of bends. The coronary is called crown. They have a lot of bifurcation. And so when you have a bifurcation or bends, the blood flow slows down. And sometimes when you drink carbonated drinks, it's acidic. And because of the acidity of the drink that you have, it flattens up the red blood cells, and you have a temporary clotting called Rollo effect. But look at this thing. High-fat diet creates high viscosity. High-fat diet, what reminds you of high-fat diet? Think about the South. What does the South do? They eat a lot of fried foods. And because of that, it's called the stroke belt of the United States. And so what happens also, if you have blood debris, you are exposed to infection, microorganism, pollutants. It destroys the glycocalyx. I showed you that before. And so look at this. You get glycocalyx disruption, and the downstream would be leading you to the clot formation here. So what did I do? I identified VLDL target 1, and then target 2 would be the inflammation, and the target 3 would be reactive oxygen species, and the target 4 would be inflammation. So I also have a drug that targets clot formation. I have identified 5 targets. So what did I do? I designed and synthesized compounds. And target 1, I synthesized that compound. I synthesized target 2. I synthesized a couple of them. Target 3, I synthesized 2. Target 4, I synthesized another 2. And target 5, I have 1 compound. But you have to understand that to be able to address the complexity of CVD, you have to combine all these compounds. And so, for example, I made 3 combos of all these things that you see on the screen. And I have A, B, up to L. And you can see the A has a combo of 226, 229, and 216, and down the line. So because we don't have a model for cardiovascular disease, the model that the government had been using is what they call a genetically engineered mouse. It's an artificial model. It doesn't work. You have to have a model that actually mimics what is happening in humans. And this is what I did. So I came up with a a mouse model, and I call it the TAP model. So I fed the mouse with hot fat diet to create a viscous blood to slow down the blood flow. And so with the reference I told you before, one of the biggest problem of cardiovascular disease is pollutant. So I took the most common pollutant in the world called PCB. And what did I do? I used this model and I look at the plaque and look what happens here. Without the drug, you have plaque. With the drug called Cambucay, which a trademark now is Embotricin, we don't have any plaque. It's confirmed by histopathology. We have to dissect them out, look at the plaque. And the plaque histopathology correlated also with the shedding of the glycocalyx. Think about this thing. So Mike told you before, we have a 4-panel biomarker that correlates with the formation of the plaque. And so so we could use this 4-panel to follow the effectivity of our drug Embotricin. So we have a breakthrough discovery, folks. This is very, very important. And so the next thing that we did is, since we know that the 4-panel is effective in following the activity of the plaque, we decided to go ahead and expand the glycocalyx, the 4-panel, and we made it the 7-panel. These are the 7 panels here. The point that I'm trying to make is, so anyway, the 7-panel, we have actually looked at and collected blood of patients that identified with different diseases. And the diseases that we come up with, and we look at the healthy. This is the pattern of a healthy. And you have Alzheimer's, you have diabetes, you got hypertension, you got stroke, you got rheumatoid arthritis, heart failure, and CHD. And you could see that there is a definite pattern of all these things. So we built a computer program, and we actually put it in a computer, and we could see that we could separate all these different diseases in a computer. So this is a breakthrough. This is going to be an important discovery equivalent to DNA fingerprint to forensics. So let's go ahead and summarize this. Embotricin in therapeutic would be the next breakthrough, like penicillin. You got infectious disease, cardiovascular disease, and the death statistic for infectious disease in 162 to 160, it killed a lot of people. You could see that. And in cardiovascular disease, we had killings as well, and we are going to be killing more people by 2030. 23 million people will get killed. Historical treatment, soybean curd. Think about it. The old days, they try everything to cure infectious disease. They use opium, iodide, mercury, arsenic, and so forth. And think about it. The mercury are toxic compounds. In the cardiovascular, we use anti-lipidic, cholesterol lowering, hypertensive, blood thinners. The predisposing factors: hygiene, poor hygiene, unsanitary environment, and over here in the cardiovascular sedentary lifestyle, high-fat diet, preservative, pollution. The causative agent over here is, in 1930, finally, they decided, they discovered that microorganisms are the cause of infectious disease. Nobody ever thought about that before. In the same token, 2010, we believe that the cause of cardiovascular disease is pollutant, xenobiotics. Xeno means outside. It's the environment. And the cause is disruption of the glycocalyx. The medical breakthrough here came in 1940 when penicillin was discovered. What we are thinking is, in the 2020s, we are going to come up with an anti-embolic drug called Embotricin. And this is going to be a first in the cure of cardiovascular disease. The whole of science is nothing more than a refinement of everyday thinking. And this is Albert Einstein. Life is defined by a blueprint DNA, but lifestyle environment defines health and longevity. And that's yours truly. Thank you.
Wonderful. Thank you, Joe. Lindsay, can you hear me okay?
Yeah, we're good.
Okay, great, great, great. So we only have 2 or 3 more slides, a couple of more minutes, and then we can get to Q&A, okay?
Okay, sounds great.
All right, wonderful. So listen, folks, what you just heard, I think, if you were really paying attention to what Dr. Tunac shared with you, we believe this could be the breakthrough of this century. And this is an extraordinary development. It's a wonderful opportunity. For us, we take this very seriously. The modus operandi of the country is to develop from the science first, science to business, not the other way around. Our focus is first the science, and now we're developing the business and business strategies around that. Specifically, our clinical strategy is to conduct 3 concurrent phase 1 studies, phase 1B clinical studies, efficacy studies, 1 within the FDA IND framework, and 2 proof of concept studies out of country, very likely in Australia, where we have a solid clinical infrastructure already in place to support us. The savings in Australia, which some of you may know, approach 50% that of US costs. We now have, I think I skipped the slide here. We're working with highly regarded providers, including Camargo Research for CRO selection and oversight, and are also building relationships with regulatory groups with strong experience in combo therapies, such as Camargo Regulatory, that assisted us in the development of what you see in front of you in terms of our timeline and exit strategy. We're currently conducting rat in vivo and in vitro studies, as well as a phase 2 ejection fraction study with Henry Ford Hospital here in Detroit. That is a translational animal study with publishable data. Finally, we expect to exit our first clinical indication, again, pre-FDA and NDA, that is prior to phase 2 studies, within 36 months. That is the plan currently. Let me get here. We now have issued patents for all 9 novel compounds that Dr. Tunac laid out for you, that he showed you. 90% of the applied methodologies, and 4 of the 7 biomarkers, which are very difficult to get patents for in the US, we have patented, used in the GlycoCardia combination that Joe emphasized. And so these are, as I said, very difficult to obtain. Our patent attorney is building what she refers to as a patent monopoly, based on the assumptions that Arterez is building a multi-billion-dollar portfolio. We've currently received 2 independent valuation analysis of our therapy versus just the 3 targeted indications that we showed you, and our IP portfolio overall. So while we're thrilled to have these early evaluation metrics, we understand the challenges ahead to accomplish our clinical objectives. That will allow us then to have the opportunity to develop solutions for other diseases, including diabetes, type 2, and arthritis. This would follow a public offering. Dr. Paul Woolley, whom developed Enbrel, some of you may be familiar with Enbrel, it's still today the leading drug against arthritis, regards our science as revolutionary, and has said he would come out of retirement to work with Dr. Tunac on a novel arthritis therapy. We're currently raising $2.5 million in a convertible debenture, which is a bridge to our Series A. The total sum we'd like to raise over the next couple of months is $2.5 million, with $10 million over 12 to 14 months, and a total need of $22 to $25 million over 30 months, which would include our clinicals. We've been approached by several institutional investors, that's VCs, interested to come in at our clinical stage. So we're not at all concerned about funding at that stage. Our concern is today in funding the remainder of our preclinical work to realize these objectives. We've established what we believe are very attractive terms for this interim raise, and we'll be happy to discuss those with any of you. We can also provide a detailed analysis and data that suggests ROI at a minimum of 12 times to perhaps as high as 80 times. And we have a sound rationale to support those numbers. Lastly, we anticipate a significant jump in valuation at each of 3 inflection points, including our pre-IND, which should be in a couple of months, submission, our IND, and of course, first clinical. So with that, Lindsay, we'd like to turn it over for any questions that anyone might have.
Great, thank you so much, gentlemen. Again, to submit questions, use your questions chat option on the right side of your screen and type over any questions that you have. I'm just gonna jump right into it, gentlemen, that's okay. So first question, Joe, how many diseases do you think a platform like this could address?
Asking you how many diseases could this address?
Oh, that's a darn good question. We are trying to cure, because the glycocalyx is the first shield, and to me, everything comes from the glycocalyx. So basically, we're addressing all chronic diseases. And I gave you a sampling before. It could cure Alzheimer's, diabetes, metabolic syndrome, arthritis. So it goes on and on, but we have to focus on CVD for now.
Great, and Joe, to clarify, with the diagnostic platform, are you suggesting diseases might be predicted or diagnosed before symptoms can become serious?
That's exactly right. We have a diagnostic. Think about the diagnostic market at this point. They're only using one biomarker for disease. It's not very accurate. It's like having a 12-man jury and you're reducing it to one. You need to have a fingerprint to be able to look at the whole picture. And that's what we're trying to do. And I told you before, it's scary because we could come up, think about it, we could come up with a fingerprint for disease equivalent to a fingerprinting called DNA fingerprint. That's scary. And we have proven that it works. We have proven that in the clinic, looking at blood from patients, that we could separate those things with the 7 marker that we have.
Perfect, great, thank you so much, Joe. So let's see, next question. Mike, you mentioned that the therapy is made up of 3 simple compounds. Has there been any analysis done to confirm that they can be scaled up easily into humans?
Yeah, we've engaged a company by the name of Cambridge. It's a very large national company, and they've done a full analysis of our compounds and determined that these would be they don't anticipate any issues to scale these up. We would like to do full GMP. That is, we would like to establish the quality of the compounds that we would be using in human because of the uniqueness and the novelty of this therapy. Immediately, that is our animal studies through chronic tox in a GMP format. However, we're perfectly able to do GLP if need be. So, but yeah, there's no issues with scaling up the compounds.
Great, thank you. And Mike, you also mentioned that your net present day valuations of your patent portfolio. Can you elaborate for us a little bit on that? And were those evaluations done by a firm with specific expertise in biopharma?
Yeah, we sought out multiple law firms in the biopharmaceutical space to help us to evaluate the value of our platform, both present and future. And we're referred to a group that only works in the biopharmaceutical space to do our evaluations.
Great. And let's see, does the diagnostic platform capitalize commercially larger than the drug or drugs?
That's a really interesting question. We really don't know the answer to that. You're asking if the diagnostics could be bigger than the drug?
I'm just reading the question that came through. That's exactly how it came.
Oh, okay. Yeah, no, the opportunity with the diagnostics is enormous. It's used twofold, both as a standalone diagnostic platform for the characterization and staging of disease, but also as a companion diagnostic for embotricin, therapeutic aid for embotricin, and any other drug that addresses any of these issues relative to the glycocalyx and inflammation and oxidation.
Can I add to that?
Yeah.
We've been seeking out cardiologists to study the science and better understand what arteries and the technologies, particularly on the diagnostic side, we're developing. And there's, the cardiologists we've talked to, at least one of them, is very enamored with the diagnostic tools. He's told us point blank that if that was available on the market today, he would be using it every day on his patients. He thinks that we've really struck on something that is hugely beneficial to his practice and adds clinical value. That's coming from a cardiologist. We've talked to ER doctors, many of them, and they view that the diagnostic tools is a patient comes into the ER with chest pains. They have no idea if that patient's suffering, is it ischemic or non-ischemic? They would have to go invasively to really figure that out. They could chest for troponin, in which case they're having a heart attack because the heart muscle's been damaged. But for chest pains, from a blood test, to be able to determine, is it ischemic or non-ischemic, so is it really happening or not, that they've all said it's a tremendous tool they would love to have today.
Great.
Lindsay, in addition to that, we've also interviewed and spoken with and shared our data with scientists and researchers worldwide who now recognize the endothelial glycocalyx as the holy grail for the study of vascular disease and solutions against cardiovascular disease. We are pioneers in this field.
Fantastic. You guys may have already gone over this as well, but are there any expected potential side effects?
You're talking about side effects?
Yes, sir.
Oh, we have, well, Mike addressed that. Did you address that? The therapeutic effect is 3 milligrams per kilogram. The toxic dose is up to 1 gram, 1,000 milligrams per kilogram. The therapeutic window is very, very high. So we don't expect any side effects.
Okay, great. Okay, so you guys mentioned you're raising $2.5 million at a $25 million cap on the Series A. Have you considered what the terms would be for the remaining $7.5 million?
This is a concurrent raise to $2.5 million. However, we recognize, and we've been approached by investors interested to consider putting in the full 10 or even the full 25 million that we need, and of course want to consider negotiating terms. We are open to negotiating the terms on the 7.5 or whatever is the remainder of the $2.5 million raise. In other words, while we're raising the $2.5 million, we'll also be talking to Series A investors about the full 10 or whatever's remaining of the 2.5, and those terms are negotiable. We are open and hopeful and looking for opportunities, investors that would like to speak with us about that.
And we also feel very strongly the $2.5 million just as a bridge to the 7.5 Series A, the equity round. That $2.5 million spend is going to help us reach key milestones, inflection points in valuation, which will drive the value of the company from a $25 million value cap into the $50-60 million range. And then we're getting validation from the market that strategy is absolutely on point. We're getting lots of agreement from biotech executives that are looking at what we're doing and how we're laying it out, and we're being very reasonable. It's all designed to give our investors, extraordinary returns at every level. So we're very sensitive to that, and we'll structure it accordingly. If someone was interested in doing the full 10 million, right as an equity round at this point, we're open to that, and we will negotiate with that.
Great, Brett. And the next question just came in, and I think this will be for you as well. Can you give us a little bit more detail on the use of the funds?
The
Use of funds?
use of funds?
Oh, sure. So the use of funds, we need to scale up the compounds, of course, move immediately into chronic tox, fully engage regulatory authority for the development of our FDA processes, including the pre-IND submission. On the diagnostic side, we need to scale up testing, remnant samples, et cetera, to kind of fine tune the scale up and development of the diagnostic kits, the proprietary algorithms, et cetera. And so all of this happens concurrently, which enables us to move toward our IND. And we can accomplish all of our preclinical objectives for that $10 million or less. And I say or less because that $10 million anticipates the worst case scenario, what we anticipate in terms of the worst case scenario, having received estimates from 4 separate CROs that have anticipated what the FDA feedback is likely to be. We've used the most expensive or worst case scenario as a means to kind of estimate timing and costs. So we know we can complete our full preclinical objectives prior to in human for that $10 million or less. The $2.5 million gets us a long way toward our pre-IND and 2 of those valuation increase inflection points.
Thank you, Mike. Gentlemen, we are running out of time here, so I do want to take a moment to thank you and to thank all of our attendees for joining us. A few of you have asked about further contact information. I will be following up with everyone individually, so if there's any additional questions, if you would like the deck or anything more, I would be more than happy to get you in contact with the team. Gentlemen, it was so great having you today. Thank you very much, and I hope you have a great day.
Thank you very much, Lindsay. Thank you, everyone.
Thank you.
Appreciate it.